Hello GPAT aspirants, welcome to online GDC classes. We have started our Pharma Thunderstorm series. In that MCQs are going to be discussed as well as theory classes are also going to be there. Please log into GDC classes app. You can watch more such videos on the YouTube also in the MCQ series. Today we are going to discuss about UV visible spectrophotometer MCQs in our pharmaceutical analysis session. And you know that UV visible spectrophotometer, it is going to have a basic instrument having more applications towards the analysis of the pharmaceuticals. Okay, so in this majorly today's MCQ series we are going to focus on the instrumental part of this spectrophotometer. That means what are the different instrumental components of this UV visible spectrophotometer, and let us see some of the important MCQs related to this spectrophotometer. Okay, so first question, if you see, the cathode of photoemissive cells is made up of, okay, cathode of the photoemissive cells, majorly the detector, whatever the detectors we are going to use in this UV spectrophotometer, that is photoemissive cells, phototubes, photomultipliers, okay, photodiodes, all these types of the detectors are going to be used, and photoemissive cells. So these photoemissive cells, these are going to have both cathode and anode. And what is the cathode which we are going to use? That is the material made up of. The question they have asked: cesium oxide, potassium oxide, silver oxide, and all of the above. Actually, whatever the cathode materials are there, they are going to be prepared from the metal oxide. So if you see the options here, all these are made up of the metal oxides. So easily you can guess that option D is the right answer. Okay. Second one, the wavelength at which the molar absorptivity is same for the two compounds is known as. That means the molar absorptivity, according to B.S. Lambert's law, this question is related, and molar absorptivity is same for the two compounds means what is that point, what is that wavelength it is going to be called it as. Whenever you are going to see for the simultaneous analysis of the any two compounds, okay, because nowadays combination drug dosage forms are majorly available, 3 to 4 drugs are also going to be included in the same formulation, because number of medications, in order to reduce the number of medications, the scientists have developed the combined dosage forms. In this you are going to use 2 to 3 compounds in a same medication. Okay, so for that purpose analysis of the combined dosage forms, it is also going to be a very critical role. For that you need not to go for the each wavelength for each substance, and you are going to see if you are going to observe any two compounds means the point, the wavelength at which, okay, so this is going to have a different lambda max, this is going to have a different lambda max, but the wavelength at which the molar absorptivity is same for the two compounds, it is going to be taken as the isobestic point, but always it is not going to be the same way, okay, but the point at which both drugs can be calculated with high accuracy and precision, that point is only to be taken, and that point it is generally called as isobestic point. Options you have seen: isocratic point, no; isocratic mode, that is related to the chromatography, both A and B, never, none of the above. Okay, so obviously the option B is going to be the right answer. Now let us see the third question. Light source for the visible spectrophotometer mostly used. Yeah, we know that in the UV visible spectrophotometer, the source of the radiation, it is going to be the one of the selective thing, and in this for UV means definitely you are going to see the deuterium lamp as the source of radiation. And for visible spectrophotometer, if they are asking here the question related to visible spectrophotometer, the light source. Okay, options: carbon arc, deuterium, tungsten, all of the above. No, actually carbon arc and deuterium, these are not going to be used for the visible radiation. Only tungsten lamp is the radiation which can give in the visible range, that is 400 to 800 nanometer. So option C is going to be the right answer. And next question, band pass for the absorption filters. Yeah, in case of the UV visible spectrophotometer, we know that monochromators are going to be used. So these monochromators are made up of dispersive elements. What are those dispersive elements? Like filters, prisms, gratings. Okay, so whenever you are going to have these filters, filters are also again two types: absorption filters and interference filters will be there. So absorption filters, what is a band pass? Band pass or band width, whatever it is there. Okay, so if you are observing any type of the entrance slit, exit slit are there, so slit widths are also very important. based on that band width is also going to be affected. And this absorption filter means definitely you can say that the bandwidth is more, okay, than prisms and gratings. So they have given the options 30 nanometer, 5 nanometer, 0.5 and none of the above. Actually D won't be there, but 0.1 means we cannot expect that. This is in case of the gratings we can expect, and 5 nanometer this is we can expect in case of the prisms, and 30 means definitely if you are going to use the filters, okay. So that is going to be the A is going to be the right answer. Next question, interference filter. Yeah, just now we have discussed about the filters. To absorbance filters will be there and interference filters. In this interference filter, dielectric space material. Yeah, interference filters means they are going to make the filters like a sandwich. So for that, in between that they are going to use a material which is having a dielectric constant, and it should be a proper metallic property also, okay. Here if you see the options, calcium fluoride, magnesium fluoride, silver oxide and all of the above, okay. And interference filters means any of these metals fluorides, okay, dielectric constant having these all these types of the materials are having dielectric constants. So in interference filters any of these metal oxides can be used, okay. For that purpose D is the right answer. Next, Fabry-Perot filter. Oh, this is one of the which type of the filter? Actually most of you have not heard about this filter name. Fabry-Perot filter is also known as option absorption filter, interference filter, both A and B, none of. Actually any of the filter it is going to be there, either absorption or interference. Both A and B could not be the right answer. D is also not there, and this is Fabry-Perot is one of the interference filter, okay. So interference filter example for this one type of the filter it is available. And next question, in UV spectrophotometer sample cell is made up of. Yeah, UV spectrophotometer we know that source of radiation is very important, then monochromator. After monochromator the light radiation enters the sample cells. So those sample cells are also called as cuvettes. These are made up of either quartz or glass. Quartz cuvettes are used for the UV radiation, whereas glass cuvettes are used for the visible radiation. Here the question is UV spectrophotometer sample cell is made up of. Yeah, for UV means you are going to use the quartz, because glass cuvettes are not suitable for the UV. Glass absorbs the UV light. So that's why option B it is going to be the right answer, okay. Quartz you can use it for both UV and visible, but glass cannot be used for the UV, right. And next question, deviation from the Beer's law occurs. Yeah, Beer's law we know that A equal to epsilon into L into C. So in this they are going to ask that A equal to epsilon into L into C, right. So this is a Beer's-Lambert's law. So deviation for this Beer's-Lambert's law occurs whenever the concentration is very high, or any type of the instrumental deviation. This is also going to the right answer. Physicochemical changes, this is also the right answer. And incomplete reaction, yeah, sometimes incomplete reactions are also going to cause the deviations. So all of the above, definitely you are going to select the option D as the right answer, okay. And next question, shift of lambda max. Oh, again the question related to the shifting of the lambda max towards the longer wavelength is also known as. So in this if you observe the lambda max, that means the graph whenever you are going to see, this is a normal bell shaped curve what we expect, lambda versus absorbance. So shifting of the lambda max whenever it is there, you are going to observe the towards any right side or left side, you are going to see shifting of lambda max towards left or right. That means towards the longer wavelengths or shorter wavelengths. So always longer wavelengths means red color it is going to be there, shorter wavelengths means blue color you are going to observe. So here the question, shifting of lambda max towards the longer wavelength is also known as red shift, okay. So distance, longer distance you can observe the red color. So in that way you can remember easily. So red shift it is going to be called as a shifting of lambda max towards the longer wavelength. Blue shift means towards the shorter wavelengths. Hyperchromic effect means actually it is related to the absorbance intensity, okay. So none of the above, no, it is not the right answer. So obviously option A is the right answer. And the next question, which one is also known as 𝘾𝙻ℎ? Okay, this is a right direct question. Actually 𝘾𝙻ℎ is a scientist name who has determined about the spectroscopy. He has done his research mostly on the electromagnetic radiation and spectral lines, how they have segregated the spectral lines. He has more research done on these spectral lines. So, 𝘾𝙻𝙎 scientist it is related to the wavelength. Options, if you see centimeter inverse, ppm, TMS, nowhere these three are related, only the option A is related to that. Next question, which of the following has the minimum wavelength, that means lesser. So, wavelength, we know that electromagnetic radiation, it is being divided into based on the wavelength, if you see. So, it is towards the higher side and it is towards the lower side. So, if you remember visible radiation, UV, IR, X-rays, microwaves, okay, like this you are going to see, and gamma rays, cosmic rays you are going to see, and highest if you observe the radio waves, okay. So, in this case, if you observe the question, which of the following has a minimum wavelength, that means towards the lower side. So, gamma rays and cosmic rays you are going to see, okay. So, obviously, actually X-rays are also going to have the lesser wavelength, but below that cosmic rays are also there. So, option A is the right answer. Microwaves, no. Radio waves, highest wavelength you are going to observe, okay. So, option A is the right answer here. And next question, which one of the following is also called as specific absorptivity? Okay, whenever you are going to observe Beers-Lambert's law as A equal to ABC, okay. So, generally you know that A equal to epsilon into L into C. This epsilon, it is going to be called as molar extension coefficient. Molar extension coefficient, this epsilon it is going to be called as, but the question related, that is specific absorptivity. Specific absorptivity means you are going to see A 1% 1 cm, then the equation becomes as A equal to small a. So, whenever you are going to see this 1% 1 cm, that is L, 1 path length 1 centimeter, concentration 1%. So, in this case only capital A is become equal to the small a, then it is called as specific absorbance value, okay. So, molecular extension coefficient means normal epsilon, you can say that. Here the concentration is not 1% and path length is not 1 centimeter. Whenever these two are 1% 1 centimeter, then only the absorptivity value is called as specific absorptivity. So, automatically B is the right answer. C, optical rotation, reverse phase chromatography, these are nowhere related to this question, okay. Next question, if you observe increase in the intensity of the, again the same related question to oxochromes and solvent effects. Whenever different solvents are used and different oxochromes are there in your sample, you are going to observe the shifting of the lambda max, okay. So, here the absorption, here they are asking intensity is going to be increased, okay. That means if you are expecting the graph like this and you are going to see the graph increasing, that means our intensity of the absorption has been increased, okay. So, always the absorbance which is higher than required, so that is hyper, okay. So, hyperchromic effect, in this way you can remember that. So, if it is lesser than the required means it is hypo, less, okay. So, this is hyper, hypo, batho and hypso. Batho and hypso, these are related to the lambda max, so shifting towards the right side and shifting towards the left side, it is going to be called as batho and hypso. That means longer wavelength, shorter wavelength. And hyper and hypo means this one. Increase in the intensity, it is called as hyper. Decrease in the intensity, it is called as hypo, okay. So, option A is going to be the right answer for this question. Now, let us move to the next question. Which one is not used as a sample cell material for the UV spectroscopy? Yeah, already we have discussed it. For the UV spectroscopy, only quartz glasses are used, okay, because it is transparent to the UV radiation. And here options quartz, glass, polystyrene and none of the above, okay. Which one is not used as a sample cell? Not used. Quartz is used. It is not used means glass should not be used, okay. So, glass which is going to absorb the UV light. Next question, which of the following solvent has the cutoff value below 200 nanometer? Yeah, UV cutoff value is also important for most of the solvents because these are also going to have some of the absorbance values in the UV region. Most of the compounds are going to be soluble in the organic solvents, that is why we are going to have the absorbance for those solvents also. So these solvent effects are also going to cause the red shift, blue shift, all these intensity variations are also going to be observed, and you should aware of the UV cutoff point also while usage of the organic solvents. And here, which of the following solvent has the UV cutoff below 200 nanometer? Below 200 nanometer, which solvent is having the absorbance? Either no, chloroform no, yeah, water is going to be the right answer. Actually, all these three are the organic solvents which are having the UV cutoff points above 200 nanometer, but water is going to have the absorbance value below 200. That's why water is the preferred solvent for the UV range. Okay, so it is not going to give you any absorbance value above 200. It is having below 200 only. So all the remaining solvents are going to have the absorbance values above 200, 205, 240. Okay, so acetone, it is going to have some at 330 also. Like that, UV cutoff values are also playing very important role. Okay, and next, band pass for grating monochromator. Yeah, previously also one question, band pass for the filters they have asked, that is 30 nanometer. Now they are asking band pass or band width for the gratings. I told you gratings are going to give you the more accurate values because the bandwidth is very less for that, 0.1 nanometer. Even if you are selected a wavelength like 250 or 240, okay, so exactly at 250.1 nanometer also you can take the reading, 250.2 nanometer also, 249.9 nanometer also. So 0.1 nanometer bandwidth it is going to be observed for the gratings. Okay, so that is the advantage. So obviously you are going to see the right answer, that is C is the right answer. Next question, if you observe B band, yeah, B band is found in which type of the compounds? Actually the question related to the absorption bands. We are going to see different types of the bands are observed in the UV radiation. Whenever UV radiation has been given to the sample, there will be excitation in the molecules. Okay, so based on the electronic shells or more number of electrons are involved here, we are going to see the different types of the bands are going to be observed based on the nature of the compounds. Okay, here the options aromatic, cyclic, aliphatic and alcoholic. So let us see for clear cut explanation on what are the different bands are actually observed. So for this B band, actually aromatic is the right answer. Let us see the clear explanation. There are 4 main bands are observed. So what type of the different bands are actually observed in this absorption spectroscopy? That is K band, R band, B band, E band. Okay, so if you remember in the alphabetical way, B, E, K, R, B, E, K, R. Okay, in the alphabetical way. So B bands, actually these are going to be observed in the aromatic or heteroaromatic compounds, typically between 230 to 270 nanometer. Okay, so these are the called as B bands, whereas K bands means you are going to see in the conjugated double bonds. Whenever you are going to observe the conjugation, okay, in the chemical structure with high intensity in the conjugated double bands, these K bands are going to be observed, whereas in case of the R bands are especially observed in the carbonyl compounds. Okay, but very low intensity because there is a forbidden transitions, these are going to be observed because of the CO compounds. And finally E band, if you see, these are going to be observed in the benzenoid compounds. Whenever benzene structure it is there, you are going to observe this strong absorption due to the benzene at 184 nanometer. Those are called as E bands. Okay, so the question whatever we have got, that is related to the B band. B band means aromatic or heteroaromatic compounds. Okay, so option A is the right answer. Now let us see the next question, the change of the lambda max. Again the same question, the change of the lambda max towards the shorter wavelength. Okay, so towards a longer wavelength it is called as red shift or bathochromic shift. Towards the shorter wavelength it is called as blue shift because blue color, shorter wavelength. Okay, so options I have given blue shift, yellow shift, green shift, red shift. So anyhow these two are not going to be there, only confusion red shift and blue shift. So towards the shorter wavelength means always blue shift. Okay, this is also called as the hypsochromic shift. Next question, light source for the UV spectrophotometer mostly used. Yeah, For UV radiation means you are going to have 200 to 400 nanometer. Always D2 lamp is the preferred one. Okay. Tungsten means it is going to give you in the visible radiation, and carbon arc definitely it is not going to give in that both ranges. So option A is the right answer. Last question, Beer-Lambert's law is related to, Beer-Lambert's law we know that it is going to give you the absorbance, right? Options are given: diffusion, no; spreading, no; absorbance, right; absorption, no. Absorption, it is not going to be the right answer. C is the right answer. Okay. According to Beer-Lambert's law, if you see A equal to epsilon into L into C, or A equal to 1% 1 cm, that means A equal to small a. You can write A equal to ABC also. Okay. So in this way you can remember very easily Beer-Lambert's law. So majorly A means that this is nothing but the absorbance value. Okay. So whenever you are talking in terms of the IR means that is a transmittance value. Okay. So but in case of the UV visible spectrophotometer, we are talking about the Beer-Lambert's law, that is A equal to epsilon into L into C, or A equal to ABC. That A is nothing but the absorbance, not the absorption. Absorption it is not going to absorb. Absorbance it is going to be the right word. Okay. So option C is the right answer. So this is going to be the last question actually. Yeah. So thank you students, thank you for attending, and more such videos you can download GDC app and watch the series, and in the GDC English channel YouTube also you are going to watch some of the MCQ series related to the all the exams related to pharmacist related exams like NIPER, drug inspector and GPAT. Okay. Thank you students, thank you for attending. We will meet in the next session.